Welcome to the deep dive. So today we have this incredibly fascinating document. It's from a series called Inside the IVF Lab. And our mission today is really to uncover what actually happens when science builds the stage, steps back, and just lets biology take the wheel in human reproduction.
Yeah, it's a great topic.
But before we get into the lab stuff, I want you listening right now to just picture IVF in your head. Like if you've seen it in a movie or, on the evening news, it is almost always the exact same visual.
Oh, absolutely. The classic microscope shot.
Right. A scientist in a darkened room, hunched over a microscope, using this impossibly tiny glass needle to inject a single sperm directly into an egg. It looks like microscopic surgery, complete
Yeah.
precision engineering.
It's a very dramatic image.
Yeah.
And frankly, I think it's a very comforting one for a lot of people because it suggests total, absolute human control over the start of life. But that specific technique, which is called ICSI, or intracytoplasmic sperm injection, is really only half the story in assisted reproduction.
Right.
Today, we're looking at a completely different foundational pathway.
Exact. Okay, let's unpack this because my understanding going into this deep dive was exactly that needle visual. You see the needle and you just think, okay, so that's how it works.
Sure, yeah.
But looking through our sources today, it becomes really clear that there is this whole other method called conventional IVF, or standard insemination. And it honestly blew my mind because the laboratory actually gives up that total control.
They do?
Like, it's not direct fertilization by a human hand, it is assisted fertilization.
Yes, and that distinction is the entire core of what we're exploring today. I mean, in conventional IVF, the laboratory creates the opportunity, but the biology absolutely has to close the deal.
Yeah.
The gametes have to do the heavy lifting themselves.
Okay, but we need to stop right here and clear up a massive misconception. If people know about conventional IVF at all, they tend to think of it as like the passive or easy version of IVF.
Like the natural way.
Yeah, like you just take an egg, take some sperm, throw them in a dish together, and go grab a coffee, right? But reading Inside the IVF Lab, it is stunningly clear that this is anything but simple.
Oh, it's far from simple. And why this should matter to you listening to this right now is that even if you never step foot inside an embryology lab, understanding this process reveals the absolute gauntlet that human reproduction actually is.
A total gauntlet.
It really is. Conventional IVF pulls back the curtain on this hidden, demanding biological sequence that we usually never get to see. Because in vivo, inside the human body, it's all completely obscured.
So let's look at the setup. Before the sperm and egg can even interact, the lab has to build this perfect environment for them to meet. The source material outlines this dish setup phase, and it honestly reminded me of preparing an ultra-exclusive VIP room for a very high-stakes blind date.
There's a perfect way to put it.
You can't just toss two VIPs into a random room, right?
Yeah.
The air quality, the ambient temperature, the lighting, everything has to be flawlessly engineered to mimic the human reproductive tract.
The VIP room analogy is spot on. But I really want to emphasize just how volatile that room is. We aren't just talking about keeping things, comfortable. We're talking about a microscopic environment that is constantly trying to fall out of balance.
And who wants to fail?
Exactly. Keeping these cells perfectly stable so they can survive outside the human body is a massive technical feat. The embryologists use something called pre-equilibrated media.
Wait, pre-equilibrated? That sounds like a highly technical way of saying they just, warm up the fluid before they put the cells in.
I mean, I get why it sounds like that, but it is so much more than warming it up. If you just warmed up the fluid, the gametes would be dead in a matter of hours.
Oh, wow. Really? Yes.
The culture media, the fluid the cells actually sit in, relies on a very specific chemical buffer system to maintain its pH. But that buffer system only works if the air surrounding it has an exact, specific concentration of carbon dioxide. So the embryologists have to place the dishes in these gas-controlled incubators hours, or sometimes even a full day, before the eggs ever arrive, just so the liquid can absorb the gases and stabilize.
That is wild. So they are essentially engineering the atmosphere of this tiny plastic droplet.
Precisely. The incubators are basically heavy-duty, heavily monitored vaults. The carbon dioxide and oxygen levels are dialed in to the decimal point. The temperature is locked at exactly 37 degrees Celsius.
Human body temp.
Right. If that VIP room fluctuates even slightly, say the pH drifts because someone opened the incubator door for too long or the temperature drops a single degree, the cellular machinery inside the egg just shuts down. The meeting never happens.
Okay, so the VIP room is perfectly engineered. The atmosphere is locked. Now it's time to introduce the VIPs. And we start with the egg, or the oocyte. This was the first major shock for me in the source material.
Cumulus cells.
Yeah, because in that famous needle injection method, the ICSI method we talked about earlier, the egg is totally stripped bare, right?
Mm.
Like the scientists can see the cell membrane perfectly.
Yes, exactly. In ICSI, the oocyte is fully denuded. All those surrounding cellular structures are dissolved away using enzymes and mechanical pipetting, so the embryologist has a completely clear, unobstructed target for the microneedle.
But in conventional IVF, the egg arrives with a massive entourage.
It does. In standard insemination, they are inseminated as cumulus oocyte complexes, meaning they retain a thick, surrounding cloud of thousands of tiny cumulus cells.
And I genuinely don't understand this part. If the whole point of moving reproduction into a laboratory is to have precision and oversight, why on earth would you leave this massive, fluffy cloud of cumulus cells around the egg?
It seems backwards, right?
Totally backwards. Wouldn't it be immensely better to strip it bare so you can actually see what you're working with? Because the source explicitly notes that with the cloud intact, the lab can't even directly confirm if every single egg in that dish is fully mature before they begin.
That's true, they can't.
That seems incredibly counterintuitive. You're flying blind.
Well, it does seem counterintuitive if you're thinking like an engineer. But conventional IVF requires you to think like a biologist. Leaving that cumulus intact isn't a lack of precision, It's a deliberate, functional choice.
Okay.
Those cumulus cells aren't just fluffy packaging. They create a highly active, natural fertilization interface.
Active in what way, though? Are they doing something other than just getting in the way?
Oh, they are actively communicating. They secrete chemical signals, chemoattractants, that the sperm actually respond to. The sperm are essentially sniffing their way through the center of that complex.
No way. That's incredible.
Right. And more importantly, that entourage provides a necessary biological hurdle. The sperm must naturally navigate and physically digest their way through this dense, sticky cloud of cells just to reach the egg surface. It's basically an endurance test designed by nature to weed out the weak.
Okay, that completely shifts the perspective. So the egg is sitting in this perfectly balanced VIP room, radiating chemical signals through its bodyguards, this cloud of cumulus cells. Now, the lab has to introduce the sperm.
Yes, the other half of the equation.
And again, my layperson assumption was that you just take the prepared sperm sample and, add it to the dish, just flood the zone, put them all in there to maximize the chances of a match. But the lab actually has to calculate an exact insemination dose.
And this requires incredible mathematical precision. What's fascinating here is we have this pervasive cultural myth that when it comes to sperm, more is always better, millions upon millions.
Right. The more tickets you buy, the better your chances of winning the lottery.
Exactly. But in the microenvironment of conventional IVF, sperm concentration is a highly dangerous variable if left unchecked.
If you think about the sheer math of this for a second, it's like a Goldilocks problem. If I go back to my VIP room analogy, let's say it's a massive ballroom.
Okay.
If you put two people in a gigantic ballroom, they might never actually bump into each other. That's what happens if the lab uses too few sperm, right?
Right. There's a vastly reduced chance of fertilization because the sperm literally just wander around the droplet and never find the cumulus cloud.
Or maybe they find it, but
there aren't enough of them to collectively secrete the enzymes needed to break down the outer layers of that cloud. Yes.
right. But then on the flip side, if you take 1,000 people and cram them into a tiny elevator, it is a complete mob scene. The system breaks down. If the embryologist adds too many sperm to the dish, you get a catastrophic failure called polyspermy.
Yes, and polyspermy is one of the absolute greatest risks in standard insemination. Human biology is strictly designed for one sperm and one egg, period.
Right.
If you flood the zone, as you put it, multiple sperm might successfully breach the defenses and enter the egg at the exact same time.
Which sounds disastrous.
It is. If two sperm get in, you suddenly have an embryo with 3 sets of chromosomes instead of 2. It's genetically chaotic, and it results in abnormal fertilization. That embryo is non-viable and cannot develop normally.
So the embryologist is doing this careful math, looking at the concentration of the sperm sample, assessing how well they're swimming, factoring in the number of eggs, the volume of the droplet, all to find that perfect middle ground.
Exactly. Enough sperm to guarantee they can breach the entourage, but not so many that it becomes a microscopic mob scene.
It's just wild.
They calculate the dose, usually aiming for somewhere around 50,000 to 100,000 progressively motile sperm per oocyte complex. They carefully pipette that specific volume into the droplet, and then they return the dish quickly to that highly controlled incubator.
So the heavy vault door closes, The lab literally steps back. And this is where the source material truly blew me away. Because the environment is set, the egg is armored, the exact right number of sperm have been calculated and added. But the actual event hasn't happened.
Not even close.
The sperm's job is just beginning. They have to run what the document essentially describes as a biological decathlon.
It is a brutal, unforgiving checklist of physiological demands. Remember, we aren't using a glass needle to bypass all of this. The lab is completely hands-off at this point. The sperm have to execute every single stage of fertilization entirely independently in that dark incubator.
Here's where it gets really interesting. I want to walk through this because reading the list of what a single microscopic cell has to accomplish on its own is staggering.
It really is. Let's walk through it.
First, they just have to survive the culture media. They have to remain viable and maintain progressive motility. They have to physically swim in a straight, strong line.
Which takes immense energy. I mean, they are relying on their own mitochondrial motors to whip their tails and propel themselves forward through the fluid.
Right. Then they hit the cumulus cells, that bodyguard entourage we talked about, and they can't just swim around them. They have to chemically and physically interact with that matrix.
Yeah, the cumulus cells are held together by this thick, glue-like substance called hyaluronic acid. The sperm actually have an enzyme on their surface that allows them to digest this glue.
Wow.
So they are vigorously swimming while simultaneously melting a pathway through this dense cloud.
Okay, so they melt their way through the cloud, which sounds exhausting, but then they hit a completely different barrier, a hard outer shell surrounding the egg itself called the zona pellucida. And they can't just bump into it, they have to successfully bind to it.
Binding is the ultimate security check. It is a highly specific, receptor-based interaction.
Which honestly made me think of a high-security keycard scanner at a sensitive government building. It doesn't matter how hard you push on the door. If the sperm's surface proteins, the keycard, don't perfectly match the exact molecular receptors on the zona pellucida, the scanner, the doors will not open.
They just bounce right off.
The sperm will just bounce off, exactly.
That is exactly how it works.
Yeah.
It's species-specific, and it requires structural perfection from the sperm head. But even if the keycard works and they bind to the shell, the door doesn't just swing open. The binding actually triggers the next phase, which is incredibly violent. It's called the acrosome reaction.
The acrosome reaction. Okay, I had to read this section 3 times. Explain how this actually works, because it sounds like a sci-fi weapon.
It basically is a sci-fi weapon. The sperm has a tiny cap-like structure at its very tip called the acrosome, and it's packed with concentrated digestive enzymes.
Okay.
When the sperm securely binds to the zona pellucida, it receives a signal to undergo this reaction. The outer membrane of the sperm head actually fuses and peels back, releasing this payload of enzymes directly onto the shell.
It's deploying a microscopic chemical drill.
Yes. It chemically digests a tiny, precise tunnel through the zona pellucida, while the tail is violently thrashing, mechanically driving the sperm head deeper and deeper into the shell.
So it melts the glue, it scans its keycard, it deploys the drill, it burrows through the thick shell, but it is still not done.
Nope, still not done.
Once it finally breaches the shell, it finds itself in a small space, and it has to physically fuse with the eulemma, which is the actual highly delicate membrane of the egg itself.
And that fusion is the point of no return. The membranes of the two gametes merge into one. The sperm essentially gets swallowed by the egg. And only after that fusion happens can the sperm deliver its genetic payload and trigger what we call oocyte activation.
Which is when the egg basically wakes up, blocks any other sperm from entering to prevent that polyspermy we talked about, and starts the massive cascade of dividing into an embryo. Viability, progressive motility, cumulus digestion, receptor binding, acrosomal drilling, membrane fusion, oocyte activation. I mean, calling it a decathlon almost feels like an understatement.
It really does.
It is a grueling, multi-stage physical and chemical gauntlet.
And that gauntlet is exactly why fertilization can still fail in conventional IVF, even when both gametes are sitting right there in the same perfectly engineered droplet.
Because if a sperm fails at literally any one of those microsteps, the entire process just stops.
Precisely. This is the inherent risk of trusting the biology.
Yeah.
You might have a sperm sample that looks fantastic under a microscope. Millions of sperm swimming beautifully.
Right, looks great on paper.
Exactly. But perhaps they have a hidden defect and lack the specific proteins to bind to the zona's keycard scanner. Or they bind perfectly, but the acrosome is defective, so they can't trigger the chemical drill.
Oh, I see.
Or they drill all the way through, but physically fail to merge with the oocyte membrane. In conventional IVF, an embryologist might come back the next morning for the fertilization check, look under the microscope, and find complete fertilization failure. Nothing happened. The sperm just swam around or got stuck in the shell and eventually died.
Which brings up the most obvious, glaring question for anyone listening to this. If conventional IVF is this biologically demanding, if it requires this insane cellular gauntlet, and it carries real risks of complete failure or catastrophic polyspermy, why do it?
That's a great question.
Why take the hard road? Why wouldn't a clinic just use the tiny glass needle, do ICSI, and inject the sperm directly into the egg every single time to guarantee the genetic material gets inside?
It's an excellent question. And it is the subject of ongoing, intense debate in reproductive medicine. The choice really comes down to the patient's specific clinical diagnostic picture, but it also heavily involves the underlying philosophy of the laboratory.
Philosophy, how so?
Well, the conventional IVF is generally chosen when the sperm parameters are adequate, meaning the lab has verified there's a good concentration, good forward motility, and no history of severe male factor infertility that would prevent them from running the race.
So if the sperm look like they have the basic equipment, you let them run the gauntlet.
Yes. And if we connect this to the bigger picture, the philosophical distinction between the two methods is profound. ICSI, the needle injection, is inherently interventionist. It completely bypasses almost all of the biological hurdles we just spent the last 10 minutes discussing.
Because the human is doing the work.
Right. The embryologist looks through a microscope, subjectively picks a single sperm based on how its shape and movement look to the human eye, paralyzes it, and forces it inside the oocyte. Conventional IVF, on the other hand, is permissive.
Permissive. That's a fascinating way to frame it.
It permits natural selection to take place on a microscopic scale.
Yeah.
Tens of thousands of sperm are competing, interacting with the cumulus cells, trying to bind, trying to penetrate.
Right.
The egg itself is playing a massive, active role in interacting with those sperm and arguably participating in the selection of which sperm is ultimately successful. It avoids the invasive physical trauma of puncturing the membrane with a sharp microneedle. Conventional IVF is not some outdated legacy method. It is a highly selective, deeply elegant biological process.
So what does this all mean? It means that when a clinic chooses conventional IVF, they are making a highly deliberate, calculated decision. They aren't being lazy. They are looking at the data, looking at the gametes, providing the absolute perfect environmental stage, and then deliberately choosing to let natural biology have the final say.
Exactly, provided the sperm actually has a fighting chance to pass the test.
They are trusting 4 billion years of evolutionary biology over a glass needle. Let's pull all of this together because the journey we just went on completely shatters that initial visual we started with. We stepped past the pop culture image of the tiny needle. We walked into the perfectly engineered VIP room of the incubator, where atmospheric gases and pH are balanced on a terrifying razor's edge just to keep the cells alive.
It's intense.
We navigated the Goldilocks math of sperm concentration, carefully avoiding the empty ballroom and the catastrophic mobbed elevator. And finally, we watched these individual microscopic cells run a brutal biological decathlon entirely on their own, melting through clouds of armor, swiping molecular key cards, and deploying chemical drills. Standard IVF isn't just throwing things in a dish and hoping for the best. It is this highly orchestrated, masterfully planned facilitation of nature.
It really is the ultimate scenario where human engineering builds the theater, but the microscopic cells still have to get on stage and flawlessly perform the play. And understanding just how demanding that play is, well, this raises an important question.
Ooh, I feel like you're about to drop something heavy on me. I'm ready.
If conventional IVF relies on this brutal, grueling biological gauntlet to naturally filter out weaker, defective sperm testing, their ability to physically swim, chemically communicate, bind, react, and digest their way through the egg's defenses, what subtle hidden evolutionary benefits might we be bypassing entirely when we use direct injection methods like ICSI?
Oh!
Right. When an embryologist just picks a sperm that looks good and forces it inside, we are skipping the entire test. Does the sheer struggle to fertilize naturally, the survival of the fittest at a cellular level, impart some unknown genetic resilience or quality to the resulting embryo? We are bypassing mechanisms of natural selection that we are still only just beginning to understand.
If the struggle itself is what guarantees the resilience of the biology, that completely reframes how you look at the entire concept of assisted reproduction. Are we helping or are we accidentally removing the most important quality control check in human existence?
Exactly. It's a lot to think about.
That is an incredible thought to leave on. Thank you for joining us on this deep dive. I highly encourage you, the listener, to keep asking those difficult questions, keep looking past the obvious pop culture answers, and stay curious. Until next time.